Hey, this is Miss Hunt for AP Bio, and I'm going to finish up our notes from yesterday. Today is Tuesday. We're going to finish up these notes, and then today you have a homework assignment to go over some more notes. So I'm going to finish up yesterday's notes, and then you have another video to watch for the second notes for this week. All right, let me view this presentation. Okay, so the second part of these notes, in eukaryotes, heritable information is passed to the next generation via processes that include the cell cycle, mitosis, meiosis, plus fertilization. We're talking about cell cycle control, and this is really big so that your cells know when to divide and then what's going on. I'm trying to figure out where to put me. All right, let me put me over here. All right, control of the cell division. Mitosis is under strict cellular control. Cells must pass through a series of checkpoints to be allowed to divide. If internal conditions are not appropriate, cell division will normally be prevented. So the checkpoints are making sure, did the DNA get synthesized in the S phase? Did it get synthesized properly? It's a checkpoint to make sure everything happened correctly. Did all the organelles double like they were supposed to? Did everything happen like it's supposed to? Which is great. So we have all these checkpoints built in. And even so, sometimes, and it's actually rare, cancers, I know everybody knows somebody who has a cancer, but overall, cancer is still a rare thing. Cancers can get through where we have a mutation in the genes and it was allowed to be passed on. And cancer is uncontrolled cell division. So when we're looking at cancers, making a new copy where it just continuously goes into making a new copy. It's basically the interphase gets cut out when we have cancer. The cell does grow and synthesize new DNA, but it doesn't spend any time really doing a job. It just replicates itself. And so that's what we get as a cancer, is just these cells that shouldn't be doing what they're doing, and they're replicating really fast. And that gives us a lump or a tumor or something for us to recognize. Internal control of cell division. Cells control cell division by controlling the presence or absence of proteins that allow for that cell division. So example is the mitosis promoting factor. So all of these little cyclins, these CDKs, these pop in to promote and allow it to go to the next cycle. This is in our textbook on 246. So if you want to click into that or flip into that, then you can look in the book and see about these different promoters that has to be able to kick in. So the kinase has to kick in order for it to go through mitosis. So each of the checkpoints, we talked about in the last unit how cells communication, and we have that process of a transduction. So basically we have a transduction happening here, and as one protein kicks onto the next protein to the next, then you can get mitosis. So it's like a checkpoint to make sure everything gets done. Progression through the cell cycle can be triggered by external signals. So here this person has a wound and they're healing back. So an external signal of a wound has kicked in, and we have a regrowth of new skin cells. So that can be an external factor that's kicked in, and the platelets or the blood that is allowed for the scab to, platelets are little pieces of broken cells that form a scab that's in your bloodstream. And those platelets will clot around a wound, and that triggers that new cells need to be made here. All right, meiosis. We went over briefly yesterday meiosis, and again, we'll go over meiosis again in heredity. So you're going to hear it a couple more times. All right. In meiosis, we produce 4 genetically unique gametes with half the number of genetic material. So here's the daughter cells that are made, and you see that they're not identical to the parent. So this is what we would call the parent cell, would be the original cell. This one has 4 chromosomes in it, and here we only have 2 chromosomes, so half the number of chromosomes. . You could think about these two as being one from your mom and one from your dad, and one from your mom and one from your dad. You don't need both of them to go into the next generation. You just need to make sure there's one orange and there's one blue going into this next generation. All right, meiosis has 2 rounds. So you basically go through what looks like mitosis, that's meiosis 1, and then you go through meiosis 2. So in meiosis 1, you're left with 2 daughter cells that are not identical because crossing over happens here in prophase 1. And crossing over is when random pieces of the chromosome switch places. And it does that to give you diversity. Nobody knows exactly why or how or which pieces are triggered. It's random, but it just happens. And that gives us diversity in our offspring. So after we go through meiosis 1, we go through meiosis 2, and we are left with 4 daughter cells. I think I said this yesterday. The correct pronunciation is meiosis. I always say meiosis in class and emphasize the E for you to remember that that is sexual reproduction and it's making egg and sperm. The correct pronunciation is meiosis, but my southern twang, meiosis and mitosis, sound a lot alike. So I usually say meiosis in class. All right, we end up with 4 daughter cells that are haploid with half the number of chromosomes. Okay, crossing over occurs in prophase 1. Homologous pairs of chromosomes exchange DNA. So these are homologous. Homo means same, and they're not identical. It's just this is your mom and this is your dad's DNA that's inside of you. Your child will only get one of these, not both of these. So which one will he receive? Will it be this one, this one, this one, or this one? Well, what happens in crossing over? A piece switches. So your child could inherit most of your father's DNA, which is kind of hard to process all this, with a little bit of your mom's DNA on it. So it just makes sure that you are unique. You are a combination of your parents' parents, and your child will be a combination of your parents' DNA as it gets passed through to generations. Another factor that gives us diversity is independent assortment. In genetics, which is next unit, we'll be doing these dihybrid squares, which are super fun, to see the chance of offsprings inheriting a trait. Which alignment, which chromosome comes here and here, what the probability is in here. This is random to see who pairs with who. There's no idea of which chromosomes get paired together. The events of meiosis and the sexual life cycle generate a tremendous amount of variation. Every organism that is produced is genetically unique. So you are a complete unique creature with your own set of DNA. A little math. All right, variations due to independent assortment. We say that there is 2N. N represents the number of homologous pairs in the chromosomes. So there's approximately 8 million possible combinations for humans. And variations due to fertilization would be 2 because you have 2 of those chromosomes. So 2 to the n times 2 to the n. So we get, what is this? Let's see, that would be million, billion, trillion, 70 trillion combinations in the human chromosomes for sexual reproduction. And then you have to think crossing over even adds more variation into this. So what this is doing is giving a chance for the offspring to be different. We'll talk about evolution at the end of this year, which is one of my favorite topics. And we talk about why evolution and why variation. And remember, this gives us that best chance. Right now, playing out, we are seeing variation with COVID. How many people get COVID really bad and end up in hospitals? How many people get it and have no symptoms at all? Different people are going to make different proteins inside of you, and your DNA is going to code for different immune systems, different ways to react to things, and that's going to cause us to have different reactions in our own bodies. So that gives us that best variation in a species. All right, that is the end of these notes, and I'll make another video later today for tonight's homework. Okay, have a good day.